everything is fine. So as we know that we are doing molecular basis of inheritance. Okay, so in this chapter, in our previous video, we learned about the uh, structure of DNA, that DNA is made up of four nitrogenous bases: adenine, thymine, guanine, and cytosine, isn't it? Even we have learned that two strands of DNA are in anti-parallel form, 5 towards the 3 direction and 3 towards the 5 direction भन्ने हामीले पढिसकेको छु प्रिभियस भिडियोमा र 5 towards the 3 direction भनेको के हो, 3 towards the 5 direction, it is already explained in the previous video, isn't it? So today we are going to study about the few information about the DNA. The first one is DNA is a double helix structure, होइन? DNA चाहिँ एउटा double helix structure हो र यो चाहिँ कसले प्रुभ गरेको हो भन्दाखेरि it was proved by the James Watson and the Francis Crick, होइन? त्यहाँदेखिको त्यसलाई प्रड्युस गर्ने को थियो भन्दाखेरि मौरिस विल्कन एन्ड द रोजलिन फ्र्याङ्कलिन भन्ने यो चारजना साइन्टिस्टले के गर्यो भन्दाखेरि दे प्रुभ द्याट DNA इज अ डबल हेलिक्स एन्ड स्टिल एनदर साइन्टिस्ट इज देयर हुज इन्भल्भमेन्ट इज देयर कि DNA इज अ डबल हेलिक्स स्ट्रक्चर हो भनेर भन्नुको उसमा चाहिँ त्यसको इन्भल्भमेन्टमा चाहिँ अझै एउटा साइन्टिस्ट छ, द्याट इज अर्गिन चार्विफ, Rosalind Franklin, Maurice Wilkin, Francis Crick, Watson and Crick. So these 5 scientists, they proved that DNA is a double helix. How? Through X-ray diffraction data. र X-ray diffraction data बाट चाहिँ तिनीहरूले के प्रुभ गर्यो भन्दाखेरि DNA is a double helix structure. Is that clear now? The next information. DNA चाहिँ we know that DNA is present in a nucleus. Nucleus भित्र हुन्छ, हैन? Nucleus भित्र हुन्छ DNA, and DNA is present within the nucleus of a cell, and it is an acid. यो एउटा acid हो. It is an acidic substance. It is an acidic substance भनेर चाहिँ कसले प्रुभ गर्यो भन्दाखेरि it was proved by Frederick Mister in 1869. र DNA चाहिँ एउटा acidic substance हो भनेर चाहिँ कसले प्रुभ गरेछ? Frederick Mister in 1869. So these are some general information that you must know about the DNA, किनकि sometimes कतिवटा क्वेसन्सहरू यो इन्फर्मेसन्सबाट पनि एराइज हुन सक्छ बोर्ड एक्जामिनेसनमा. ओके, सो नाउ वी आर गोइङ टु स्टडी अबाउट द साइलेन्ट फिचर्स अफ द DNA. एक्चुअली डिएनएको स्ट्रक्चर पढिसकेपछि त्यो साइलेन्ट फिचर्स पढिरहनु पर्दैन, यु क्यान राइट युर सेल्फ अल्सो, बट अल्सो एकताल हेरौँ, होइन? फर्स्टमा के रहेछ भन्दाखेरि टु स्ट्रान्ड्स अफ DNA को दुईटा स्ट्रान्ड्स जुन चाहिँ छ, ओके न, टु स्ट्रान्ड्स अफ DNA कुरा बुझ्नु भयो भने इज मेड अफ two polynucleotide chain, एउटा polynucleotide chain and another polynucleotide chain. So that means two strands of DNA is made up of two polynucleotide chain, and यसको ब्याकबोन चाहिँ के रहेछ भन्दाखेरि यो जुन चाहिँ साइटमा भएको केले बनेको हुन्छ, साइटमा बीचमा तिम्रो को हुन्छ? नाइट्रोजनस बेस. यहाँनिर को हुन्छ? तिम्रो sugar and the phosphate. That means what is the backbone of DNA? DNA को backbone के रहेछ? Sugar and the phosphate. Whereas in the center you will get the nitrogenous bases. Is that clear? So first characteristics of the DNA is made up of two polynucleotide chain in which sugar and phosphate is a backbone, whereas nitrogenous base is inside. कुरा बुझ्नु भयो? nitrogenous base त केमा हुँदो रहेछ? बीचमा हुँदो रहेछ. ओके, नेक्स्ट वान. सो मैले अस्ति भनिसकेको छु हाम्रो प्रिभियस भिडियोमा कि the structures of two strands of DNA are in 5 towards the 3 direction and 3 towards the 5 direction भन्नुको मतलब they are in an anti-parallel form. That means two strands of DNA are in anti-parallel form. Is that clear? अब यो कुरा भयो. Number 2 feature. Number 3 features के रहेछ भन्दाखेरि we know that DNA is made up of 4 nitrogenous base: adenine, thymine, guanine, cytosines. So adenine paired with the thymine with double bond, and guanine paired with the cytosine with triple bond. Is that clear? So that means third features ki bha, DNA has 4 nitrogenous base in which adenine paired with the thymine with a double bond, guanine paired with the cytosine with triple bond. Okay, next one is, last feature is that DNA is in a right-handed fashion. भन्नुको मतलब यस्तो स्ट्रक्चरले बसेको हुन्छ, यसो डल्लो परेको हुन्छ, हैन? र the pitch of the helix is 3.5 nanometer. nanometer भनेको चाहिँ के हो भन्दाखेरि यो जुन चाहिँ strands of DNA जुन चाहिँ छ र यसको यो जुन चाहिँ साइटको छ नि, यो जुन चाहिँ यो साइटको छ नि, यसलाई हामी पिच भनेर भन्छौँ, र यो चाहिँ कतिको हुँदो रहेछ तिम्रो? 3.4 nanometer. भनेपछि बीचमा को छ? नाइट्रोजनस बेस. Is that clear? So that means the structure of DNA is right-handed fashion, and the pitch is made up of 3.4 nanometer. And next one is, well, in the previous video we have learned that जब अब adenine, thymine, guanine, cytosine, adenine, thymine, guanine, cytosine, adenine, thymine, guanine, cytosine, 1 2 3 4 5 6 7 8 9 10, जब 10 base pair पुग्छ हैन, when 10 base pair reach, so after 10 base pair DNA will take a turn. That means how many base pair are there in one complete turn? 10 base pair. That means 10 base pair in each turn. That means it means that कि DNA को structures मा चाहिँ each turn मा कतिवटा base pair हुँदो रहेछ, 10 वटा base pair हुन्छ. So there are 10 base pair in each turn. So okay, this is about the general information as well as the features of the DNA. So in the next video we'll learn more about this chapter. Thank you very much.